Good morning. What a real pleasure to be able to welcome Dr. Deanna Barch as our distinguished visiting lecturer for the month of October. One thing you need to know is that when I finally was recruited and accepted the position here, one of the first people I reached out to was Deanna, as a colleague and as a friend and as someone I admire, who has played such a key leadership role in the development of really modern psychiatric neuroscience research as we think about it today in 2018, but also as someone I knew who herself was a department chair at her home institution at WashU. And I emailed her, told her I'd accepted this position, and asked if there was any sort of advice or guidance she could give me as I started to think about and prepare for the role. And in her very characteristically generous way, because Deanna is not only noted for her kind of scientific and intellectual kind of contributions, but also as being an incredibly generous colleague. She sent me some own work she had done when she first became department chair, which was a self-study she had done in her own department, and that was very inspirational to me as kind of a first set of steps that I could think about and take as a chair. So I'm very grateful to you for doing that. And again, it's this, and the self-study was thorough, it was detailed, it was unflinching, but its underlying theme was how do we develop our faculty, how do we create a culture which nurtures and supports faculty to be the best that they can be, and again, very, very characteristic of the approach she's taken in her professional life. Dr. Barch is the Gregory Couch Professor of Psychiatry and the chair of the Department of Psychological and Brain Sciences at Washington University in St. Louis. She received her undergraduate degree from Northwestern, completed graduate school at the University of Illinois in Champaign-Urbana, and then did an NIMH-sponsored postdoctoral fellowship at Western Psych Institute and Clinic at University of Pittsburgh, where she worked with our colleague Angus MacDonald, my colleague John Cohen, Cam Carter, kind of a whole coterie of us who are all kind of from that same generation. Dr. Barch's contributions to the field of psychiatric neuroscience research are almost too numerous to count. I'll tell you, she's now become really, in this phase of her career, really a thought leader. She's editor of the she was editor of the Cognitive, Affective, and Behavioral Neuroscience Journal. She's deputy editor at Biological Psychiatry. She's on the editorial boards of Schizophrenia Bulletin, Current Directions in Psychological Science, Journal of Abnormal Psych, Clinical Psychological Science. She was the immediate past president of the Society for Research in Psychopathology. She's on the DSM-4 Revision Committee. She's on the steering committee for the NIMH Research Domain Criteria Initiative and is, or just was recently, a member of the NIMH Scientific Council. She's done, again, groundbreaking research for many years funded by NIMH, NARCED, NSF, the Dana Foundation, et cetera, and the recipient of several awards, including the American Psychological Association's Distinguished Scientific Award for Early Career Contributions to Psychology and the Joseph Zubin Memorial Fund Award. She's also a fellow of the Association for Psychological Science and a member of ACNP. We're going to hear about one of her current threads of research, but I think summing it up, we could say that essentially she has been deeply interested in the interplay among cognition, emotion, behavior, brain function, neural circuit function, to understand how these things interplay to create the behavioral deficits that we see in depression and schizophrenia. And she's particularly interested in sort of the developmental aspects. How do these things interplay over the course of development to create risk for these diseases and then expression of these diseases? She uses functional MRI, structural MRI, and cognitive neuroscience methods to look at sort of the disturbances in these processes. And she's also become interested in recent years in the ways in which adversity, such things as disparities in access to health care, poverty, stress, shapes early brain development. And indeed, we heard about some of the work she's been doing in collaboration with Joan Luby when Joan was out here last year. So I will turn it over to Dr. Barch to thank her again for coming here, and she will be speaking to us about her work in connectomics and psychosis.
Well, thank you for that very generous introduction, and thank you for inviting me. It's wonderful to be here. I partly feel like old home days, many friends and colleagues who are here, and it's been wonderful so far to speak with people. What I want to do today is to talk about one of the lines of research in my lab that has been focusing on the relationship between functional connectivity, brain system organization, and risk for psychosis and cognitive deficits across the lifespan. And I want to disclose that I don't really have any disclosures. But I also want to start by thanking the many, many colleagues and students who have contributed to this work. Anybody who does work in psychosis and functional connectivity knows that it is team science for sure. And many people have contributed to the work I'm going to be talking about today, and also to thank the generous funders for the work that I'm going to be talking about today. So I'm going to start out because maybe not everybody in the room is a believer, maybe not everybody in the room knows about functional connectivity, but to make some arguments about why you might be interested in looking at functional connectivity if you are interested in trying to understand the neural contributions to risk for, development of, and maintenance of psychopathology. And today I'm mostly going to focus on psychosis, but I think this, the arguments that I'm going to make could easily apply and do apply to other forms of psychopathology as well. So let me start by giving you some reasons why I think we should care about functional brain connectivity. Most of the forms of psychopathology that we're interested in are ones that we know have a genetic contribution. None of them are entirely genetically determined by any means. There are very important environmental contributions, including adversity, but also family influences and other things. But we know that most forms of psychopathology have an important genetic contribution. And so one of the things that we know about functional connectivity is that it is also heritable. And that's a characteristic that you would want if you were trying to understand pathways from genes to risk for psychopathology. Now you might say, how do we know that functional connectivity is heritable? Well, I've had the privilege of working on a project called the Human Connectome Project. And as part of that project, we recruited a very large sample of young adults, and we saturated the sample with families that included either monozygotic or dizygotic twin pairs, as well as other siblings. So many of our participants came in families of 4, where there was a pair of twins plus 2 of their siblings, both monozygotic and dizygotic. And we know that monozygotic twins essentially are identical genetically. I don't say completely identical because there's things that can happen that make you slightly different, but MZ twins, monozygotic twins, are much more genetically similar than dizygotic twins. And so to the extent that things are more similar in monozygotic twins than dizygotic twins, that is suggestive of a genetic contribution to that characteristic. So we were able to look at the heritability of functional connectivity as part of the Human Connectome Project. This was an analysis we did early on looking at 15 pairs of monozygotic twins, 12 dizygotic twins, 71 pairs of non-twin siblings, and then 8,417 unrelated pairs, because you can take any one of these data sets and pair up all the unrelated people as an estimate of how similar even unrelated people are. And then you can look at patterns of connectivity. There's many different ways you can do this. We happen to do it by parcellating the brain into different brain regions, identifying the patterns of correlations in spontaneous brain activity, and then asking how similar those patterns of correlations were across individuals. You can literally sort of, correlate the patterns of correlations. And what I have here plotted is the correlation magnitude of the similarity. So higher values are more similar patterns of functional connectivity. And what I have plotted here are monozygotic, dizygotic, siblings, and unrelated individuals. And what you can see is that the similarity of the monozygotic twins is significantly greater than the similarity of the dizygotic twins, which is in turn more similar than the siblings, who don't share an in utero environment, although they are as similar genetically as the dizygotic twins. And then the siblings are more similar than the unrelated individuals, suggesting also some environmental contributions. Others have since replicated these kinds of findings in even larger samples of our twins from the human connectome. So we're fairly convinced that there is evidence for the heritability of functional connectivity patterns, and this also doesn't seem to simply be secondary to structural similarities, which we also know are heritable. If you kind of dig in and say, well, what are the most heritable connections in the brain? So variation in the heritability among different patterns of connectivity or between particular brain regions. Interestingly, where you see the most heritable edge is in the connection between one of the regions of the default mode network, the posterior cingulate, and I think Sarah will be very interested in that, and parts of the inferior frontal gyrus that are thought to be important for impulse control and emotion regulation. So this is actually the most heritable connection in the whole pattern of brain connections. And interestingly, this is actually what we would call a negative edge, meaning that they're anti-correlated. So as activity in the inferior frontal gyrus goes up, activity in this region of the default mode network goes down, which is a pretty typical pattern that we see. So evidence that it's heritable, this is a positive attribute if you're interested in mapping between genes to risk for psychopathology. Another important thing is that it changes across the course of development. So I think another thing we know about most forms of psychopathology is that they really have their roots earlier in development, that they don't spring out of nowhere late in life or, even in early adulthood, but instead evolve over the course of development, and that for many forms of psychopathology, you can see early indicators much earlier on in the individual's life. And so you, again, might be interested in a neural mechanism, a neural contributor that also shows developmental changes that might give us clues about what are the mechanisms that might be contributing to risk that emerges and evolves over the course of development. So we now have an extension of the Human Connectome Project called the Lifespan Project, that is designed to really look across the course of life. And prior to the current project, we did a big pilot study with 100 people from kids down to age 8 up through 80, and we looked at connections among brain regions. And to do so, we used a particular approach to identifying brain regions. And we use a parcellation approach. We refer to it as the Gordon parcellation because the guy who was the first author of the paper, his name is Evan Gordon, that actually looked at continuities and discontinuities in the patterns of connectivity across regions of the brain. So what I have plotted here are individual regions where all the voxels within that region tended to show very similar patterns of connectivity, but very different patterns of connectivity from adjacent regions that are colored in a different color. So for example, all the parts of the brain here in this red area tended to show very similar patterns of functional connectivity that were different than these regions in blue or light green or other green here. You can do that, and you can identify a bunch of regions in the brain that show similar patterns of functional connectivity. And then you can ask whether those different regions aggregate into brain networks that span the brain. And in fact, they do, and many, many different approaches to parcellating the brain and then identifying networks have identified very similar networks. So for example, the ones in yellow here are referred to as the frontal parietal network. They have been identified in almost every way of parcellating the brain and looking at brain networks. The darker purple here is the cingulopercular network. And I'm going to come back to both of those later on in my talk. And in red is the default mode network that includes, dorsal medial prefrontal cortex and posterior cingulate and precuneus and other regions. So this is pretty replicable across individuals, across data sets, and even to some extent across development, so that the sort of basic organization shown here is present even early in the development. But you do see changes across development in the magnitude of the interrelationships among brain regions within a network and between different networks. So for example, what I have plotted here is age in years in our, we call it our preliminary lifespan sample, so going down to age 8 up through age 78, I think was our oldest. And what I have plotted here is the strength of the connectivity within that cingulopercular network. And what you can see is that that's actually one that declines over the course of aging. I actually have a prediction or a hypothesis that when we actually have the many more subjects that we will have in the full project, we'll have closer to 1,200 people spanning this early age range that will actually see more of a kind of an inverted U-curve where it actually goes up a bit into adolescence and then comes down. But we definitely see this decline with aging, and it's not accounted for by artifacts like motion and other sorts of things. We also see changes in the connectivity between networks. So again, age, and here we have the connections between the default mode network and the ventral attention network, which we again see declining over the course of age. But we see other networks that actually show an increase in connectivity over age. So this is the connection between the visual network and the somatomotor body network. So these are networks that of brain regions that activate when you're presented with visual information, and then also do like touch or feel or actions. When I first saw this, I didn't believe it. I was like, why would that be going up across the course of age? We've now replicated it in a different data set, so we think it's real. We could have an interesting conversation about what this might reflect. But you see both increases over age and decreases over age, so that we're seeing the brain connectivity in the human brain evolving as a function of development. Again, something that might be interesting if you're trying to understand risk for psychopathology. And then lastly, I would argue that it's important to have measures that relate to behavior. In the end, psychopathology is about behavior experience of humans, right? And we want to be able to link our measures to some interesting aspects of behavior that we think are relevant to understanding the experience or the symptoms of people with different forms of psychopathology. So we and a million other people have looked at the relationship between functional connectivity and relevant behaviors. I'll give you one example from the Human Connectome Project, but you could easily find many others in the literature. And this was analysis spearheaded by Steve Smith, where he took about the first half of our participants, it was about 490 some odd people. He did a different approach to parcellating up the brain called ICA, Independent Components Analysis, identified a bunch of different brain regions and networks in the brain, and then he used a technique called canonical correlation analysis that lets you take a set of predictors on one side and then a different set of predictors, in this case a whole host of behavioral metrics, and ask what are the interrelationships between this set of connections among brain regions and all these different behaviors. And importantly, he found a first canonical variate that really mapped this particular pattern of connections to higher scores, better scores across a range of behaviors. And higher scores here were mostly about better things, about life satisfaction, substance use, psychopathology, cognitive performance. And the way it related was that all the red connections here are stronger connections among these brain regions predict better performance here, and the blue ones are more anti-correlations or negative connections predict better performance here, okay? If you then, again, try to unpack that and say, well, what happens if we take the aspect of behavior that had the strongest loading on that variate? And the thing that had one of the strongest loadings was fluid intelligence, matrix reasoning. This is a measure that actually shows all kinds of interesting relationships to real life experiences, like how well you do in school, whether you have a job, like real things about people's everyday lives. And if you look at the correlation there, so this is the, oops, go too fast, sorry. There we go. If you look here, this is the weighting for fluid intelligence, the performance of the participant, and then this is how similar their pattern of brain connectivity was to that pattern I showed you. You see this very strong prediction of fluid intelligence, suggesting that there is a relationship between how one's brain is organized and how one performs on these kinds of important cognitive tasks. Okay, so it's heritable, it changes across the course of development, it seems to relate to important aspects of behavior, which we think are important characteristics for helping us to understand psychopathology. So we have been over now probably 5 to 10 years engaging in a line of research trying to look at particular networks in the brain that we think are important for cognitive control and cognitive function, and try to understand how they might be disrupted in psychosis, how they might help us understand cognitive deficits in psychosis, and whether they actually might be part of the risk pathway to the development of psychosis. So let me start sort of temporally at the beginning, but also with what I think is one of the more basic questions we need to ask about functional brain connectivity, which is the basic organization of brain networks the same in people with schizophrenia or not? Because if it's wildly different, right, that might have very different implications for the kinds of questions we can ask or what the mechanisms might be that are driving differences in functional brain connectivity. So this was work that was done by my recently completed graduate student, Dov Lerman-Sinkoff, and he used graph theory approaches to try to look at identifying brain networks among individuals with schizophrenia and healthy controls, and then asking whether they were similar or different. So let me show you this first. So this is a set of healthy controls. These are brain regions from one of those parcellations, like the Gordon parcellation. And he ran it through some fancy graph theory statistics called InfoMap, and it identifies brain regions that organize together. And what hopefully you can see here are some pretty similar brain regions to what I showed you in the previous map. So in red, we see the default mode network. In this kind of blue here, we see the frontal parietal network. In this kind of darker red here, we see the cingulopercular network. We see this repeatedly in study after study. And the question is, if you do this in schizophrenia, does it look like this or does it look wildly different? Well, okay. Apparently my thingy doesn't want to work today. Okay. Oh, now it's really happy. Speed talk. And we'll see if this wants to work here. So what you'll see here is essentially the identical pattern. We see the default mode network in pretty much the same place. We see the frontal parietal network. We see the cingulopercular network. We had to squint very hard to see any differences in this organization of brain networks amongst our individuals with schizophrenia versus controls. And when we squinted really hard, we saw very minimal differences in a few subcortical connections. So the basic organization seems to be very similar. I think a number of other people have seen this. If we do the same thing in the first-degree relatives of patients with schizophrenia, we see something similar where the basic organization seems to be quite intact. Here's another way of looking at it. So here now we've just organized all those regions into rows from 1 to 264 regions. And then you do this InfoMap analysis a whole bunch of different times at different thresholds, right? Including different, I'm only going to include correlations that are 0.2 or above, or 0.1 or above, or 0.05 or above. And you look at how consistently that brain region gets mapped to that same network. And so this is being very, very conservative, only taking into account the very strongest correlations, and this is less conservative. And you can see that many of these are very consistently mapped to the same networks, no matter how conservative or non-conservative you are, and that this pattern, again, looks essentially identical amongst the individuals with schizophrenia, right? So starting from the perspective that it's not a wildly different brain organization, but that it may be more subtle variation in the strength of connectivity among regions in a brain network or between different brain networks. All right. So there are additional tools that you can bring to bear from graph theory to try to understand brain organization. And graph theory is a very interesting approach because it wasn't at all developed to be used with brains, right? It was developed to look at the organization of things like social networks or plane hubs or roads and highways in different countries. And so it's really this sort of agnostic mathematical approach to saying, how do things work together? What is the strength of connections among things? And you have to start by thinking about nodes and edges to kind of understand how graph theory works. My wonderful graduate student, Julia Sheffield, made this example for me. So you can think of nodes like cities, right? So here's Chicago, here's St. Louis. If you haven't been, we have this really great arch. Come see us. This is Nashville, where she happened to be from. So those are nodes, and you can think of edges in many different ways. One way to think of them is the sort of size of the highway that connects those cities. So, you might have a very big highway, 55 is a pretty big highway, 64 is a pretty big highway, but you might also have cities that are connected by a country road or, a dirt road, right? So they're going to differ in how many different lanes are in the highway, making it, easier or harder to get between those cities. You can also think about the physical distance between the cities, right? So, St. Louis and Chicago are closer than St. Louis and Minnesota. So if you're thinking about physical distance, you might say that there's a stronger edge between St. Louis and Chicago than there is between St. Louis and Minnesota, right? So graph theory can be used with any of these different kinds of metrics of sort of strength of connections between nodes, right? But that is not what functional connectivity represents, right? So functional connectivity is not about how close two brain regions are, and it's not about the physical, size of a connection. If you get into more structural connectivity, you might start getting into the sort of physical size. It's really about how similar are the patterns of brain activation over time in those different brain regions. So you can still apply all the graph theory metrics to that measurement, but I think it's important to keep in mind that we're not talking about physical distance or kind of physical size of connections. But it does involve thinking about nodes as brain regions, as I articulated, and then the strength of correlations or similarity in patterns of connections. Once you do that, you can adopt some of the metrics from graph theory that are designed to tell you something about the organization of that brain region. So, for example, one metric that you can pull out of graph theory is referred to as efficiency or global efficiency. Formally, efficiency is the inverse of something called path length. You might say, well, then what is path length? Well, path length is how many different edges or connections do you have to travel down in order to get from one node to another node. So in this example here, right, if I want to get from this yellow node to this red node, the shortest way to do it requires me to travel down 3 paths. 1, 2, 3, right? There's no shorter way to get there. So the path length between this node and this node is 3, and efficiency is the inverse of that, 1 over 3. The path length between this node and this node would be much longer, right? Because you have to traverse many more edges, right? If you're going to look at global efficiency of a network describes essentially the average path length among connections in that network. And it is thought to potentially be an index of the sort of efficiency of information transfer among brain regions within a network. Right? So that is typically how it's interpreted in the literature and how we think about it. So higher global efficiency in a brain network, we interpret it as more efficient transfer of information among that regions in that network. Right? And we typically think of that as potentially being good for whatever function that network supports. Feel free, by the way, if there's any questions or anything that I'm saying is not clear, feel free to interrupt me and ask questions. Now, we've been interested in looking at particular brain networks that the literature suggests are important for cognitive function. And so the 3 that we have been particularly focused on are the frontal parietal network. And so these are the regions in yellow. They include dorsal frontal and parietal regions. They are thought to be important for cognitive control and task representation, and in particular more adaptive aspects of cognitive control, helping to change regulation as you're involved in a task, being able to maintain goal representations and change them as you need them. We've also been interested in the cingulate opercular network, which is the anterior insula and the dorsal anterior cingulate, and in some studies also parts of dorsal parietal or angular gyrus. And this is also a network that is engaged by a whole host of different cognitive tasks. Some have argued that it's involved in sort of stable task control. We've argued that it may be more important for kind of indexing conflict that arises as you're engaged in cognitive processing, and then signaling to other brain regions that they need to upregulate their activity in order to help you perform better on the task. So it's a network that gets very active, for example, when you make errors or when you are experiencing challenging cognitive task conditions. And then lastly, we've also been interested in the default mode network. The default mode network is this sort of mystery network that people are very interested in, but we don't know exactly what it does. One prominent theory about what it does was put forth by my colleague Mark Rakel, who has argued that it's important for attending to internal states and kind of regulating your attention to the internal state of the individual versus sort of external task demands. Others have also argued that it's very important for sort of self-assessments and sort of making assessments about attributes about yourself and your internal state. As I kind of alluded to before, it shows a pretty different pattern of brain activity during cognitive tasks than the frontal parietal and the cingulopurcular networks. It actually tends to decrease its activity when you're engaged in cognitive tasks. And many people have argued that that sort of push-pull relationship is really important for effective cognitive function. That if, in order to be able to be good and on task, you need to be able to sort of damp down activity in the default mode network and damp down attention to your internal state so that you can be attending to the task at hand. So these are three networks that we've consistently looked at in relationship to psychosis and cognitive function in psychosis because of all the literature on their role in cognitive function. Okay. All right. Now, in addition to thinking about networks as a whole, you can also think about hub regions in the brain. And you might say, well, what's a hub region? Well, hub regions are thought to be regions of the brain that might be particularly important, not just for information transfer within that brain network, but for helping to sort of transfer information across brain networks or to sort of organize the relationship between brain networks. There's a number of people who've argued that, why does this keep doing that? There we go. That there are a number of regions in the brain that serve as hubs. So one is the dorsal anterior cingulate and the bilateral anterior insula. People have argued that these are hub regions that help communicate. Some have argued between the default mode network and like frontal parietal and cingulopurcular networks. Others have argued for one of my favorite brain regions, the dorsolateral prefrontal cortex, as a hub region, with the idea that it might help actually arbitrate among a whole bunch of different brain regions, and in particular might be important when you need to rapidly your sort of new task representations, when you encounter new things in the world that you've never encountered before, that the dorsolateral prefrontal cortex might be important for like rapidly reconfiguring brain networks to do this novel task. Okay, so these might be hubs in the brain. You might say, well, how do you assess how hub-like they are? Well, one way of doing that is another metric from graph theory called the participation coefficient. And the participation coefficient is simply a measure of how many different networks a particular region connects to, depending on whatever threshold you set for your network connections. So in this example here, our red node has a connection with this little network over here, this network over here, and this network over here, right? So it's connected to 3 different networks, as opposed to this one here, which is really only connected to that one single network, right? So this red node would have a higher participation coefficient than this yellow node, and I might have an individual where this node is connected to 10 networks, and another individual where this same node is only connected to 2 networks. And so that one individual would have a higher participation coefficient for this node than another individual. And so this may be more about information transfer across networks rather than information transfer within a network. Okay. All right. So my student Julia Sheffield started working with data that we had among individuals who were already diagnosed with schizophrenia, looking at whether or not efficiency in those networks that we were interested in related to cognitive function among individuals with schizophrenia. And what she found, well, she used data actually from a collaborative project that I have with Angus MacDonald and others called Centrix. And this was data from a multi-site imaging study that we did. And as part of that imaging study, people performed a number of different cognitive tasks. They did a processing speed task, they did a verbal learning task, a cognitive control task, an episodic memory task, and a visual integration task. We could have a whole other talk about what each of these tasks measures and kind of what specific cognitive function they assess. But we actually decided to take a different approach in this study and to say, can we kind of pull out a more general metric of cognitive function? So we did a principal component analysis and took the first principal component, which was kind of the major mode of variation in cognitive function, and asked whether global efficiency of these brain networks could predict individual variation in this more general aspect of cognitive function. And indeed, what we found was, if we looked at the frontal parietal network, so again, these are these regions in yellow, what I have plotted here is this cognitive factor score, so higher is better. And then what I have plotted here is global efficiency of these networks. In red are the individuals with schizophrenia, and in blue are the healthy controls. All right, my pointer is like, what is the word? It's like haunted today. And what you can see is that actually across both the folks with schizophrenia and the healthy controls, we see that higher global efficiency of the frontal parietal network is associated with better performance on these cognitive tasks. And we see something similar if we look at the cinguloopercular network. So again, the regions in purple are the cinguloopercular network. In this study, we actually see that it relates to cognitive function in both the individuals with schizophrenia and healthy controls. We didn't see a differential relationship in the individuals with schizophrenia. Now you might say, well, maybe this is true just across the brain, right? Like the better your brain is hooked up, the better your cognitive function, and it doesn't really matter what specific networks you're looking at. Well, turns out we don't see this at all with the default mode network in this study. We also looked at other comparison networks. So for example, we looked at the auditory network, which has about the same number of nodes as the cinguloopercular network, and we don't see it there. So we see at least some evidence of this relationship being more specific to the frontal parietal and cinguloopercular networks, which are the networks thought to be most consistently involved in cognitive function. We then looked at hubs and whether or not participation coefficient of any of those hubs I noted, so dorsal anterior cingulate, bilateral anterior insula, or the dorsolateral prefrontal cortex, was also related to cognitive function. And what we saw was that the anterior insula, at least the right anterior insula, also showed that relationship, such that better cognitive performance was associated with higher participation coefficients. And again, the insula and the dorsal anterior cingulate have been argued to maybe be important for sort of that push, kind of arbitrating between that push-pull relationship between the default mode network and the frontal parietal and cinguloopercular networks, right? But again, you'll note in this particular study that relationship was present both for the folks with schizophrenia and the healthy controls, suggesting that this wasn't a unique relationship to the individuals with schizophrenia. Now, we've done lots of work in schizophrenia, but we know that schizophrenia is not the only form of psychopathology that involves cognitive deficits. We know that there are other forms of psychopathology that also involve psychosis that also seem to show similar patterns of cognitive deficits to what we see in schizophrenia. So we could include schizoaffective disorder in that, and also people who have bipolar disorder with a history of psychosis show very similar patterns of cognitive impairment, although not always as severe as what we see in schizophrenia. So we have wanted to ask whether these kinds of relationships are really unique to sort of psychosis, or whether there might be something more transdiagnostic. So we followed this up with data that our colleagues who ran a different project called the B Project generously shared with us. So this is Carol Taminga and John Sweeney and Macheri Keshaman, among others, who had collected data from a pretty large sample of individuals with schizophrenia, schizoaffective disorder, and psychotic bipolar disorder, and they collected some resting state functional connectivity data and graciously shared it with us. And so Julia also led a series of analyses with this data. So she did something very similar with cognition as in the previous study. They had administered a range of cognitive tasks, so she did a principal component analysis, pulled out the first principal component, and then you can also see the individual tasks for those who are interested in more specific things. And what you can see here is that we replicated a pattern that had been seen in the literature before, where we see impairments among all of these groups, but that it's somewhat graded, with the biggest impairments among the individuals with schizophrenia, followed by schizoaffective, followed by psychotic bipolar disorder, compared to healthy controls. And you see that same pattern across all of the different tasks, although the overall magnitude of impairment does vary a little bit across cognitive domain. But the very similar pattern is there. So we see this sort of transdiagnostic, albeit graded pattern of cognitive impairment across kind of the spectrum of psychosis. What about global efficiency in these same brain networks? So let me first show you the mean levels of global efficiency. So here is our friend, the frontal parietal network, the cingulopurcular network, and then here's our comparison network, the auditory network, and here's whole brain efficiency. So a couple things should stand out. So first off, whole brain efficiency, if anything, you actually see that whole brain efficiency is increased in psychosis. And this is, we are not the first people to see this. It has been argued that this reflects sort of a little bit of a breakdown in the modularity of brain networks across the brain in psychosis. But perhaps more related to our hypotheses, if you look at global efficiency of the cingulopurcular network, we see that overall it's decreased among all the individuals with psychosis. We actually do not see reductions in the frontal parietal network, and we didn't see it in the auditory network. So it seemed to be more specific to the cingulopurcular network. And because papers often become better when you send them out for review, when we got this reviewed, the reviewer said, no fair, we know you had hypotheses, but you should really test every brain network. So we ended up testing every brain network, and we found that this relationship was very specific to the cingulopurcular network, and the only other network that showed this reduction was a subcortical network involving striatal regions that one might also expect to be altered in schizophrenia. We then looked at how global efficiency in the cingulopurcular network related to cognitive function transdiagnostically, and here what I have plotted is the cognition measure. Here's cingulopurcular global efficiency, so better cognition, higher global efficiency. Blue here is the healthy controls, and here's the 3 patient groups. And here we actually saw an interaction with diagnosis, such that only among the 3 patient groups did we see a relationship between global efficiency and cognition. And in all 3 of the patient groups, worse global efficiency was associated with worse cognitive function. And this actually mediated in part, the diagnostic effects on cognition were mediated in part by the cingulopurcular global efficiency. Okay? So not just in schizophrenia, it's present also across the spectrum of psychotic disorders. And here with a larger sample size, we started to see some evidence for stronger relationships to cognition among individuals with schizophrenia, schizoaffective, and bipolar compared to healthy controls. Now, all of these are people who already have the illness, meaning that we don't really know whether this kind of alteration in global efficiency, particularly in the cingulopurcular network, is sort of a consequence of living with and experiencing psychosis, or the medications that people might take, or a host of factors that sadly come along with having a major mental illness, or whether maybe some of these things might be present prior to the onset of illness and might actually be part of the risk pathway to the development of psychosis. So to try to address this, we've actually started looking at individual differences in psychotic-like experiences in more general population samples who have not yet been diagnosed clinically with schizophrenia. So the start of this was actually in the Human Connectome project, where we happened to include a questionnaire that included some questions about psychotic-like experiences. Particularly, do you hear voices that other people can't hear? Do you see things that other people can't hear? Do you behave in unusual ways? I know that's not very specific. And do you have strange ideas that other people don't understand? Again, I know that one's also not very specific. I got ahead of myself. So that was the mediation model. Okay, so what do we see in this general population? So this is, again, a higher order cognitive ability factor. We took the range of cognitive tests that we administered in the Human Connectome project, did a first principle component, extracted it out, measured cingulopurcular global efficiency, and looked at the interrelationship. And what you see in this general population sample is, again, that higher cognitive ability is associated with stronger cingulopurcular global efficiency. Then, if you look at PLEs, or psychotic-like experiences, you also see that people who have greater psychotic-like experiences also have worse cognitive ability globally. If you then look at singular-opercular global efficiency in psychotic-like experiences, you also see that people who have higher levels of psychotic-like experiences show reduced singular-opercular global efficiency. And if you put this all together, what you see is that the relationship between psychotic-like experiences and impaired cognitive ability is mediated by reduced singular-opercular global efficiency. So those people with higher levels of psychotic-like experiences show reduced singular-opercular global efficiency, worse cognitive function, and that singular-opercular global efficiency seems to mediate that relationship. Even in this general population sample, right, that it includes people who have not yet or may never be formally diagnosed with schizophrenia, okay? Suggesting that this extends beyond the kind of clinical spectrum really can't be secondary to things like medication effects or this experience of living with a severe mental illness. Now, in this sample, right, these are people who are, yep, That's a great question. So one thing is that the people who score high on these psychotic-like experiences, many of them don't have some of the other associated symptoms of psychosis, so they may not have like the negative symptoms that often are very debilitating. They may have other resilience factors, so maybe they have, really supportive family networks. So that's been a really interesting question, because some of these people function quite well, even though they have, pretty consistent levels of psychotic-like experiences. But they seem to be different in this, like not having the other constellation of symptoms, not always having as much distress associated with them. And some of the psychotic-like experiences are a little bit more on the border, so they might be like, I hear someone calling my name, but I'm not necessarily hearing full-blown voices. Absolutely, absolutely, yeah. That's not specific to a general population sample, yeah. But again, I think some of the psychotic-like experiences that these folks have just aren't of the same extremity that we see in clinically diagnosed. It really is more that schizotypy flavor, right? Like if you've ever worked with people who have more kind of schizotypal features, it's definitely on that spectrum, but it's not the full-blown voices that, have repeated phrases that they say to you and so on, or like really well-formed delusions. It's more kind of odd beliefs. We see a lot of people who have beliefs that are more culturally accepted, but are a little bit more unusual. So beliefs about psychic experiences and things that are well accepted in some communities, but are maybe a little bit out of the unusual norm. So, okay. Now, though, I should point out, right, most of these are 22 to 35-year-old adults. The vast majority of these people are never going to go on to develop schizophrenia. So we don't really know, is this just sort of another part of the spectrum? Is it part of risk? To answer those questions, you'd really need to start much younger and understand how these things evolve over the course of development. And so here I've had, I'll call it the good fortune, Katie can tell me whether that's the right answer, to be involved in another large-scale project called the Adolescent Brain and Cognitive Development Study. And that study is recruiting, when we're done, we'll have recruited almost 12,000 kids, they're all ages 9 and 10. And one of the things and it's a very general population sample, we're trying to roughly match the demographics of the general population. So it's not pre-selected. I wouldn't fully call it epidemiological, but it's a pretty broad sample. And one of the things that we're very interested in, among the many other things that the study will look at, is the temporal relationship between the emergence of psychosis and the use of substances, particularly substances like cannabis. Because there is suggestion in the literature that use of cannabis might actually be causally related to the emergence of psychosis. But this is very controversial, and there are very few samples that actually have assessed kids before any exposure to substances and any development of psychotic symptoms, potentially development of psychotic symptoms, to understand that temporal relationship. So because that's one of the goals of the ABCD, we actually included a much more in-depth measure of psychotic-like experiences in the ABCD. So what we used was something called the modified PQB, which is the psychosis questionnaire brief. This was a measure developed by Rachel Lowy and colleagues. I don't know why my computer is doing this. I'll tell you about it until it decides to come up. So this was a measure that's been used in originally in young adults and adolescents, 21 questions that assesses a range of psychotic-like or psychotic experiences. And they had already been in the process of modifying that for use with children, because of course it's much more difficult to ask about psychotic experiences in children, because some of the normative experiences that children have could easily be called psychotic. So imaginary friends. How many people had an imaginary friend when they were young, right? Some of you did, you may not admit it, but it's very normative to have imaginary friends up until like age 8 or so. And then you start to see the frequency in the general population of imaginary friends go down. But by the time you're 9, 10, 11, 12, it's not very typical to have an imaginary friend, right? Kids are also a little bit more suggestible. You know, they worry about scary clowns in the closet, especially after they've seen the movie It, right? Am I going way over? Okay, good. Okay. So they modified it. So I'll just give you an example. Original question, have you had experiences with telepathy, psychic forces, or fortune telling? And the question we now ask is, did you ever feel like you had special or unusual powers? You could make things happen by magic. You could magically know what's inside another person's mind. And we can get 2 scores here. We can just get the number of times a kid says they had one of these experiences, but we also ask about the distress associated with it, because there is evidence that distress is a really critical differentiator of what sort of not really a psychotic-like experience versus something that's bothering children. And we ask about a whole host of things. I am saying this in part because this is all publicly available data, so any of you who are interested in psychotic-like experiences can go download this data and do what you will with it. We do follow up to some of the questions and ask the kids what we mean, so that you can sort of differentiate between when it's, you know, kind of a typical normative fantasy versus something that's more bothersome. Just as a funny anecdote, we started this study right after this movie It came out about the scary clown. There's a whole lot of discussion of It in one of the questions from kids who were like, my mom and dad let me see It, and now there's a clown outside my window. So don't let your kids see It. So the data I'm about to show you is from 4,524 kids that we acquired in 1 year, and about half male and female, pretty demographically diverse. Just to convince you that this is measuring something real in these kids, here is the average distress score as a function of family history, and what you'll see here is that the kids who have a family history of psychotic disorders show increased distressing psychotic-like experiences, and that's not true as a function of family history of depression or mania, so consistent with what you would expect. We also see that it relates nicely to cognitive function, so higher distress scores are associated with lower scores on a composite measure of fluid intelligence and a composite measure of crystallized intelligence, and that's true even when you account for other things that we know predict cognitive function and psychosis, like family history of psychotic disorder, income to needs, those sorts of things. And if you break it down, you actually see that it relates to the same kinds of cognitive impairments that are often seen most strongly in psychosis, like processing speed, working memory, vocabulary, and reading, right? So some converging evidence that this seems to maybe be a valid measure of psychotic-like experiences in kids. We've now just recently looked at how it relates, again, to singular opercular global efficiency. And once again, we see this is 4,500 kids, so there's a lot of dots in here. But we see a significant inverse relationship, similar to what we saw in the adults, similar to what we see in patients with schizophrenia, where higher PQB distress scores, so more distressing psychotic-like experiences, are associated with reduced singular opercular global efficiency. This is a longitudinal study, so what we will be able to do now is track these kids over time and look at whether PQB scores and network organization actually predict things like whether they show sustained distressing psychotic symptoms over time, who develops more clinically diagnosable psychotic disorders, and I think also interestingly, how does this interact with cannabis use, right? Does it predict who is going to use cannabis? Does it predict response to cannabis or use of any of other host of substances? All right, so I will wrap up there by hopefully I've convinced you that we have some interesting tools for measuring in vivo functional connectivity. We can use them with a wide range of ages and populations. There are all kinds of interesting other analyses coming online all the time that I think will be more informative as we go forward about the nature of functional brain connectivity. As I argued, it changes over development, it's heritable, it relates to important things that I think make it relevant for understanding psychopathology. And we think that it's starting to help us understand the pathway or some of the risk factors to cognitive dysfunction and psychosis across the spectrum from sort of more healthy population to clinical populations. We will see whether it will be a risk factor for the development of more clinically diagnosable psychiatric disorders over the next 8 to 10 years as we follow these children longitudinally. But we still have much to learn. We really still need to understand exactly what functional connectivity reflects in terms of the underlying sort of molecular mechanisms that drive that. We know it is related to structural connectivity, but it is not just structural connectivity. It's not isomorphic with that. So we need to do much more to understand what the mechanisms are there. So I will stop there, and thank you.
Do we have time for some questions?
Yeah.
Amazing talk, Deanna. That was incredible. You've been very busy.
Or my students have.
So most of the data we're looking at were resting state data. Is that right? So some of the people in the audience might be thinking about these networks as targets for intervention. And oftentimes, the goal of intervention is not to improve resting state, but to improve active cognition, engagement in cognition. So I'm just wondering, how generalizable you think these networks are to when the brain is probed with a cognitive task. So, for example, like a conflict task, like the Stroop or the DPX. Would you expect, those bars for the singulo-opicular network to change in terms of the group differences if you're using an active probe like that?
So we've done that too, not in these samples, but in a different sample, and we see pretty similar. So there's two ways you can answer that question. You can say, does it predict, how related is it to connectivity during task, and how related is it to activation during task? So we've done the analyses looking at connectivity during task, and we see very similar patterns of deficits during connectivity during task in these same networks as in rest. That's not to say that there aren't changes in the strength of connectivity as a function of task. There's some interesting changes. Some things go up, some things go down, but we see the same pattern of deficits in schizophrenia during task as we do during rest. We have not looked at whether, but this is an interesting thing to do, whether individual differences in functional connectivity during rest predict individual differences in activation during task, but other people have, and they see pretty strong connections. In fact, there's been some really interesting analyses, some of them done by a guy named Mike Coles, who was a postdoc with Todd, my husband, but he's done a little bit of schizophrenia stuff, but he's done some really interesting stuff looking at whether sort of flow during resting state predicts activation during tasks and finding some interesting things, but he hasn't then applied that to understanding schizophrenia. So I think that's a really interesting set of questions. I suspect they're going to relate. I think these networks are organized the way they are for a reason, and it predicts how they are able to respond to challenges in the environment, but we haven't done some of those further analyses.
So are we wasting time collecting task data?
I'm not going to say that. Somebody would knock me down on the way out the door.
So that may be, this may be a different version of the same question, but does that relate to the observations made in the past by Weinberg and others about increased drive and demand cortical activity on, say, working memory tasks in at-risk or prodromal relatives of patients? Is it the same question or a different?
I think it's a related question. I mean, I think it does raise, I mean, Joe Callicott and Danny and others have argued, and I think there's now data to suggest that part of what's driving that is that what you see is that at sort of lower levels of cognitive demand among people at risk for or with psychosis, you actually see greater activity, and then when they sort of hit their capacity, which tends to happen sooner than in healthy controls, then it drops off. So you see patterns of both increased activity and decreased activity depending on the nature of the cognitive demand. And, I think it's a very interesting question as to whether these patterns of disrupted network connectivity might predict who is going to show that kind of pattern, right? So if your network isn't as well organized, maybe it's harder to kind of get it going to engage in these cognitive tasks. Again, the general population analyses would suggest there's a relationship, but we haven't done that specific analysis in psychosis yet.
I'm wondering whether you showed a lot about disconnection of a network leading to cognitive dysfunction. Were there networks or connections where hyperconnectivity caused worsened cognitive function? You might expect like a perseverative or habit-driven network, for example.
We have looked and looked and looked in I cannot tell you how many studies, and we have never seen that. And the place you would think would be default mode network. I mean, I have been looking, Mark Rakel has been bugging me to look at this since like 2002, and we have looked and looked. We just never see it. Some other people have seen it. Whitfield Gabrielli has seen it in some small N samples. My former graduate student, Alan Antichevich, has like once maybe seen it. We look and we just don't see it. You might have thought, but we don't see it. We do see it in other forms of psychopathology. So we do see more evidence for that in depression with like increased default mode connectivity being associated with depression, which kind of makes sense when you think about ruminative sorts of things. But we have just never seen it in psychosis. And we have tried and tried. Yeah. All right.
I noticed that while significant, all of the, or most of the correlations that you showed had really small effect sizes, meaning the amount of variance explained is really low. Yep. I wonder how, and I really don't mean this as a criticism, but like, how do you think about that? Like, is it, do you just think it's like sort of imperfect on both variables, or is it like some kind of additive effect across networks? Like, what are your thoughts, basically?
No, that's a great point. And I think this is actually a particularly relevant point as we get into these giant samples, 4,500 kids, right? I can make anything be significant, right? And believe me, it is. I think it is partly that, we're not going to find the big smoking gun, that it's just one contributor among many. So I think that is a big part of it. I think that really is the effect size. I don't think it's like bad measurement and, now many of those studies, I will say, not the human connectome, but like the BSNP study is like 6 minutes of resting state data, like not much data. But the human connectome is an hour of resting state data, so I can't really say it's it's that we don't have enough data. And the ABCD is more like 15 to 20 minutes, so decent amounts of resting state data. One of the things, so there's some really interesting work that's been starting to go on in not our lab, but we're hoping to adopt it, looking at more individually defined networks and showing that actually increases the magnitude of your correlations with the behaviors of interest pretty significantly. Because all of these analyses are you take a group template and then you simply slap that on the individual. And we know now there's increasing evidence that that the basic organization is there in most people, but that there are really interesting individual differences. And that if you can incorporate those individual differences into your analysis, you might increase it. So that's on our list of things to do over the next couple of years, is to change our analysis stream to start taking that into account and seeing if we can increase those, because they're consistent. We see them across studies, but they are not big effect sizes. Yeah. You had a question?
Yeah, I was wondering if you could maybe try to give us a little more insight into what the meaning of some of these measures like shortest path length on resting state networks is, given that, I was thinking the underlying signals are being sampled once a second, once every 10 seconds. So the time scales we're talking about are multiple seconds.
Yep.
And I had in mind also throughout your talk, Michael Cole's result that the less reconfiguration networks have to undergo between
Right.
resting state and task, the better.
Right.
And I guess I had one question, which that hasn't been studied, I'm guessing. But
Yeah.
if you could just, think a little bit about, or out loud about what all of this means for how we should be thinking about how the brain works and doesn't work in pathology.
Right. So that's a very deep question, right? In terms of like, how do these networks get configured and what drives the individual differences, right? So, some of it, some people have argued that some of it is basically a kind of a Hebbian idea, right? That regions that fire together wire together. So Steve Peterson and others have argued that might be one of the driving forces behind these patterns of functional connectivity, that, that as you are, operating in the world over the course of development and you are engaging in various tasks, the extent to which these brain regions tend to co-activate drives the development of these patterns of functional connectivity. Although the mechanism of that coordinated activity, I think, is still not at all well articulated. That may be true, but there's not much experimental evidence that shows that to be true. Like people have tried to do some things where you look at if you have people engage in a repetitive action or train them on something, do you actually see an increase in the functional connectivity of brain regions? This has been done a little bit more in the motor domain, and you actually do see some evidence of sort of increased connectivity. So people have taken that as evidence that maybe some of this, activating together, firing together means that you wire together. That cannot be the whole story, though, because we have also done, and this is a loose we because it's really my colleagues, neonatal brain imaging looking at functional connectivity, and you see some proto-organization of these networks present right at birth. And then there's even people now, like Mariah Thomason, who's been doing fetal imaging, resting state fetal imaging, and sees some proto-evidence even in utero. So that suggests that it can't all be post-birth experience, for example. So maybe some of that is having to do with, some, genetically driven principles of brain organization. Now, Mike's observations about the less reconfiguration you have to do, the better you do on task, basically, I think is a really interesting one, because it suggests, again, that, maybe that could be a principle of, behavior. The more you are engaging in these tasks and doing well, the more you're, wiring up these regions, and they're showing this pattern of functional connectivity even at rest, and you don't need a lot of reorganization to get to task. But again, we don't have an experimental manipulation that shows that. So great questions. There are people who are trying to tackle this in animal models and monkey models to try to better understand, what exactly does this mean? Because it's not just based on monosynaptic structural connectivity. You see these functional connections among brain regions that, are pretty widely separated and are not, directly connected in terms of sort of white matter connectivity. So it's got to be something beyond that. Okay. Yeah,
you had already answered my question, but I was going to ask about if individual subject differences are being kind of masked over by putting everybody into a single template space.
Yes, I think they are. And that's going to be the next wave of much more difficult analyses to do. I think you had wave where you're...
You had mentioned the possible relationship between marijuana use and onset of psychosis and possible changes in cognition and connectivity. So if that relationship exists, what would it look like and how might that happen? Any leads there?
Oh, that's really speculative, right? I don't know. I mean, I could tell you what I would predict, right? Which is, if you think, so it depends on your model of the relationship between cannabis use and psychosis. So we have a paper that's about to come out where we looked at twin models of the relationship between psychotic-like symptoms and cannabis use, and tried to look at whether whether it was sort of a shared genetic predisposition versus an exposure. And what we saw there was that, at least in our analyses, which were 2 big twin samples, one from the human connectome and one from an Australian twin registry, that a good chunk of that relationship seemed to be shared genetic predisposition, so that those who were genetically predisposed to have psychotic-like symptoms were also using cannabis. We saw some hints of an exposure effect on top of that, we can't say that our data prove this, but we're sort of intriguingly consistent with the idea of sort of a double hit idea, that there are things that make you more likely to both be predisposed to psychotic-like symptoms and to use cannabis, but that among those people who are at risk, using cannabis may have more of a negative effect than people who are not at risk, right? So both predisposition and exposure effects. So if this all worked the way we thought it might, what you might see is that people who have poor cognitive function, reduced single-auricular efficiency, are both at risk for using cannabis, more likely to use cannabis, and more likely to have psychotic-like symptoms. But maybe it would be the case that people who had psychotic-like symptoms or reduced single-auricular efficiency would have a stronger reaction to using cannabis. So that cannabis may not have the same effect in everybody, that it maybe, this is all very speculative, right? Maybe it would have more of a sort of, more of a negative effect on increasing psychotic-like symptoms or worsening cognitive function or efficiency amongst those at risk than amongst those not at risk. All speculative, no data to support this, right? But that would be one way it could manifest itself. I do think there's a lot of evidence in the literature for shared predisposition, but then there's also some evidence for the exposure effect. So I think my personal view is the most likely explanation is this: it's both predisposition, and then once you use it, it's going to be more taxing on a vulnerable system. Yeah.
This was a great presentation. Thank you so much. This is a political question, though. The percentage of the population that is at risk for what you were just talking about, because after the November elections, Minnesota is likely to get recreational marijuana. Mm-hmm. So that's one question. The other one is a sort of a hypothesis question. You didn't talk about rapidity. You talked about functional and anatomical connectivity, but the rapidity with which, so like P300 data shows that the affected population has a slower P300. Mm-hmm. So this is metaphorical. I got a scooter and you have a Ferrari, and a tsunami is coming.
Yeah, I'm going to get out of there quicker.
Yes, and I'm going to take a shortcut, which as soon as I get there, I'm recruiting a part of my brain that says, oh, you made a big mistake. So I was wondering whether there's some relationship between your findings, connectivity, processing speed, and synaptic efficiency.
I think it's a really interesting hypothesis. We definitely see a relationship to processing speed, but not only to processing speed. However, I will say that there are some papers starting to come out. One of my previous students has a paper coming out looking at structural connectivity and processing speed, where they do see a relationship, and she's made a similar argument that, maybe it is something about sort of the speed of information transfer that's being degraded if these networks aren't well organized. And so and there's a whole host of things that could do to cognitive function. There's a lot of models about that. So I think that's a perfectly reasonable explanation, although I would say that all of our cognitive effects related to functional connectivity don't seem to be solely accounted for by processing speed, right? So it may be partly that, but maybe not just that. The cannabis question is a really interesting one. That's actually one of the reasons ABCD got started, was because there was concern that legalization is coming, and we just don't have data one way or the other, right? So in the psychosis field, there's 2 really strong camps. There's a camp, Robin Murray and others, who vehemently believe that cannabis is causing psychosis, and that the severity and rates of psychosis are worse in East London now because of stronger cannabis. he's argued very strongly for that. There are other people who argue, no, the data does not support that at all. They support only a predisposition model. There's no evidence for causal effects of cannabis on psychosis. So that's part of the reason for the ABCD study, is to get longitudinal study about this, because we don't, it's not that we have data that we're not paying attention to, we just don't have data, right? Because we have not been allowed to do the research. So we don't know. Good, bad, nothing, we just don't know. So, yeah.
I think many of the findings you reveal seem to suggest that the singular or percular network is kind of like reflecting the overlap between psychotic disorders and the general cognitive ability. Mm-hmm. And then I know, like, for general cognitive ability is one of the most, one of the traits that is like most shown to relate to functional connectivity. So I feel like the struggle for me in my research is like whether I control for that when I'm studying for psychosis, and if I don't, if maybe all the functional connectivity correlates that I found with psychosis will just be, what is like reflecting that covariation with cognitive ability. So I kind of just want to hear about your philosophy of approaching that.
Yeah. So I think my philosophy is that the cognitive ability is a key, is kind of a core aspect of psychosis. So I don't like to think of controlling for it, because I think of that as a really important component of the illness, although it's certainly not present in everybody. It's not a symptom of schizophrenia. Sophia mentioned that I was on the DSM-5 committee. We had a lot of conversation about whether to include that as a symptom, and we ended up not, because it's not specific to schizophrenia. It's present in, many forms of psychosis. But if you control for it, you may be controlling for like a really critical aspect of the illness. Having said that, I think if you want to know, does something relate to something else irrespective of that, if you control for that, you can ask about whether that relationship is due to the cognitive, ability, or kind of goes around the cognitive ability. And I think knowing which of those is true is always important. So we do always try to kind of look at it both ways, but we don't think of it as like a nuisance or an artifact, but rather as like an important part of the story. So, good question.
Great. We'll pause here. It's been a fantastic presentation, a great discussion. Thank you again, Deanna.
Older gentleman who gave me this is hearing. Yeah. Is it his? He left. Oh, there he is. Okay.
You have been disconnected from the meeting.